I wanted to we're gonna get started. Welcome everyone, and thank you for coming today. It's my pleasure to introduce our seminar speaker, Marissa Sandoval, who's a grad student in the population biology program. So Marissa came here from UC Berkeley, where she did her undergrad in molecular environmental biology. One of the many programs that we have there at UC Berkeley. And from that time, Marissa was working, doing research in the lab with Noah Weinman. They do a lot of work on insects, and Marissa was, as far as I can see in her CV, she was very, very keen in studying insect biology, insect behavior, which is a good reason for joining population biology in our lab. And so since arriving here, Marissa has been really interested in understanding both the genetic and the chemical basis of communication in insects. And the Orchid bee system that we study in the lab is great for that. So Marissa has been tackling this really challenging question of understanding chemical signals and what's the language, what's the information containing these signals in the context of mating, which is very exciting and interesting for the system that we study, but also broadly for the study of sexual selection, chemical communication, animal communication. And her work is really challenging, but at the same time, very, very exciting because she's onto something really, I think, big, which is trying to understand the meaning of these signals and how we can understand them better. And that requires a lot of patient dedication and careful experimental design, which is all the things that Marissa's doing. Without further ado, Marissa, take that away.
I'll just wait for some folks to sit down. Okay, thank you so much, Santiago, for that introduction, and thank you all for being here and coming out today. I'm excited to talk to you a little bit about the research I've done and what I'll continue to do while here in Pop Bio. So let's get started. We're all biologists and likely got interested in biology because of the diversity of forms and different traits and function that we see all around us. And it's been said that variation is the raw material for evolution to act on. And there are some really classic examples that we're all fairly familiar with, such as the Darwin's finches and the size of the beaks, as well as in a color phenotype in rock pocket mice, matching the substrate that it lives on. And both of these types of traits exhibited variation and were important for fitness in the context of survival. But variation in traits isn't only important for survival. Another component of fitness, that being reproduction, traits often play a role in reproduction. And secondary sexual traits, those that aren't directly involved in copulation, are often a way for selection to act on trait variation. And so this is nicely perhaps encapsulated by Charles Darwin's frustration with peacocks, and that the sight of a peacock's tail, whenever I gaze at it, makes me sick, is something that he had said. And he couldn't fathom how such an organism could exist with this clunky tail all around, but it impresses mates. And so secondary sexual traits are important for recognizing conspecifics. And so I'm really happy to be following up on Rick's talk last week, where we talked about plant communication potentially, and he had introduced us to the concept of a cue, which is a stimulus that elicits a response from the receiver, but wasn't intended, so to speak, as a means for communication. Whereas a signal is a stimulus that was evolved for the means of communication, from sending information from a sender to a receiver. And in this case, throughout the talk, we'll be talking about quarters and choosers. And so here we have a little cartoon of a sender sending a signal to receivers, and one being a conspecific. And so to put some biological context to this, a nice example in the literature are the Tungara frogs, which maybe some of us are familiar with. These frogs have characteristic whines and checks, and males will call to females, but also there are eavesdroppers. And so frog biting midges, as well as frog eating bats, also use that call, but it's not like the frogs are trying to get eaten, right? So for the purposes of the talk, we'll just be talking about signals. I may accidentally slip up and say cues, but I really mean communication. And so these types of signals vary in complexity as well as sensory modality. And mate choice research is a really cool intersection of all these different disciplines coming together from, different psychology and sensory biology and, all of these various disciplines in behavior too. And some differences and sensory modalities that have been explored are colors, like in visual cues, and signals like in cichlids that exhibit different colors, and that being important for mate choice, and auditory communication, such as in the frogs, and we're all listening to bird songs every day, and even kind of tactile substrate-borne cues and signals such as spiders that communicate via substrate vibrations. And the elephant in the room kind of has been olfaction, because though it is an old means of communicating, so to speak, it's been neglected as one of the sensory modalities, or has been less explored, let's say. And all organisms from microbes to us use it, perhaps unbeknownst to us. And some examples in the tree of life are birds have uropigeal glands, in which differences in communication, differences in chemistry are shown between the sexes, as well as swordtail fish, though, for visual communication of the length of the tail, they also have differences in pheromone composition between the sexes. And within insects, chemistry is a primary means of communicating. And exploring olfaction as a signaling modality can be tricky, partially because of our own biases that we bring as humans and researchers. We're very visually oriented, as well as, more prone to listening and auditory cues. And and when we look at this image of a bee visiting a flower, we're often paying attention to its physical behavior of how it approaches the flower, and maybe hearing the bzz sound, we're cued in on that, but less so in like seeing the lens through her eyes, so to speak. Even our vocabulary is a little bit more visually oriented, and we can experience and know what it's like to get really excited smelling certain aspects of a flower. And so what also makes it difficult are that a lot of these pheromones and chemicals are internally synthesized. And so extracting them means perhaps like lethally, killing the insect, which we often don't want to do. And even when we do know, what types of compounds are involved in these organs, then it becomes a challenging question of asking what is important. Once we know, usually in these glands, there are a bunch of different compounds, and figuring out what's the most important and like what is actively conveying information can be tricky. And at least, with other systems, we can manipulate the signal in a perhaps more feasible way by artificially elongating a swordtail fish, or doing callbacks of artificial calls of frogs, or putting dollar store feathers on birds and seeing how they respond. But with chemistry, it's a little bit tricky. And so you can use genetic modification, and that often can be done, but it's also tricky, but it's not impossible. And so though challenging, we know from the wealth of mate choice literature that a powerful system would involve quarters, if we're interested in this, which we are, who exhibits, quarters would exhibit secondary sexual traits in the first place, and then perform those behaviors in an experimental setting. And this is often hard to do in animal behavior because animals don't like living in captivity. And so we want our quarters to court, but we also want our choosers to choose, since it's no good if you have your quarter courting and your choosers they're not responding. And lastly, it would be ideal if we can confirm matings. And this is often really tricky. Sometimes animals are promiscuous and they mate multiple times. And sometimes we have to resort to association times. How much time does an organism spend on this side of the cage where the signal is present versus over here where the signal is absent? But sometimes interest doesn't mean choice. And so when you can confirm choice, that's really powerful. And so I'm here to tell you that orchid bees are a really compelling system to explore all of these exciting questions in behavior and mate choice for all of these reasons. So the males themselves do happen to have secondary sexual traits, and they aren't intrinsic to the organism. Orchid bee males, a lot of you are familiar with, will actively collect from the environment different oils from environmental sources and concoct a sort of perfume to impress females and to attract females. And uniquely, each species has a kind of unique perfume blend. We can manipulate the signal without disrupting behavior, and I think this is really important. Often you will, to kind of look at response, we douse insects in different, chemicals and see how, nest mates respond, but they spend all this time grooming themselves and perhaps not doing the behavior that we want them to do. So being able to precisely manipulate a signal without affecting behavior is really key. Females aren't promiscuous. They only mate once in their lifetime that we know of. And we can also confirm this through dissections of spermatheca in females. And so if you haven't seen an orchid bee collecting perfume from an orchid, it's quite exciting. That's where they get their name from. And so this is a Gonkora orchid, and you see a male doing this really nice behavior of using his forelegs to kind of brush up all this oil and then put them in his hind legs for later. And they don't just collect from orchids, they also collect from a bunch of other environmental sources. So here's a video that one of our colleagues at a botanical garden in Florida took of all these different males collecting from lotus flowers. And so naturalists have long thought that, this type of behavior was associated with courtship, but it's really difficult to see actual matings in the wild, like surprisingly. And it wasn't until last year that one of our colleagues was able to, kind of investigate this experimentally by doing a cool project in South Florida, where a species, Euglassa dilemma, has been introduced from Costa Rica. And so it involved acquiring naive males that had never collected perfume before and naive females that had never mated before. And he inserted perfume into one male and left the other male without perfume and saw the response. And so what was neat to see is that the presence of a perfume didn't mean that the male wouldn't exhibit courtship behavior. So on the right side of the slide, you can see the control males are those without the perfume, the treatment males, those with the perfume, and they spent just as much time trying to court females. So the presence of the perfume wasn't necessary for courtship. And this is what that looks like. Males will find a perch and then kind of waft off the perfume with the wings. But when it came to matings, all the females basically mated with males that had perfume compared to those that lacked them. And so this was really exciting. People have been interested in orchid bees for a long time. And so having, clear results like this now laid the foundation for all these other really interesting questions regarding this type of behavior and the information conveyed by the perfume. So that's where I come in with the research questions that I'm really interested in. The first being, as I had mentioned, that each species has a unique perfume blend. Does that facilitate mate recognition and preference for conspecific mates? And we're still going to be talking about the species that was introduced in Florida, Euglassa dilemma. Secondly, moving from between species to within a species, how does the perfume composition and complexity influence mate choice? And lastly, at the individual level, to what extent among different individual perfume bouquets in males, how much do they differ when the environmental conditions, the perfume sources remain constant. And so throughout the talk, I'll be using these little icons in which, the two colored bees, or we're talking about the first research question, the one colored bee, we're talking about the second research question within a species, and the individual bee, we're just talking about among individual males. Alrighty, so for the first research question, do species-specific perfume mixtures facilitate mate recognition of and preference for conspecifics in Euglassa dilemma? We can go back to the cartoon in which we had already talked about recognition. And preference is when there are multiple options and you're deciding between them. So you already recognize that these options are, in this case, males of the same species, and then deciding amongst them. And there's some argument whether or not this happens sequentially, but preference is, a decision that is relative. And so these are the two species that we're working with, Euglassa dilemma and its very recent sister, Euglassa viridissima. And they were cryptic and only described in 2011. And at least, again, as visually oriented organisms, they look quite similar. Euglassa dilemma has 3 teeth on its mandible, and Euglassa viridissima in the orange has 2 teeth on the mandible. But what was, the stamp of why they were different was, of course, the perfume. These species co-occur together in the Yucatan Peninsula, and viridissima comes up on the west side of Mexico, and dilemma extends down to Costa Rica. And so work that our collaborators and a previous grad student, Santiago's, had done really nice work looking at the different perfumes throughout the allopatric and sympatric ranges, and found that indeed the perfumes were distinct from each other. And this was really neat because it was confirming something that we didn't know before. And so a lot of my work in sampling was in the similar regions that Philip had already visited. And to answer this question, it started with that, collecting perfume in the regions of symmetry. That way the bees had access to the same sorts of environmental sources to craft a perfume to begin with, and pooling the samples together to then use for 2 choice mate experiments. And lastly, using microsatellites to confirm which male was a lucky one of 2. And so I went to Mexico with a colleague, another graduate student who's been working with me on this project, and we collected perfume from as many bees as we could in the time. And we identified them in the field with a little hand lens looking at their mandibles. And after collecting perfume, we took little aliquots to then confirm that RDs were correct. And so what you're looking at here are 2 chromatograms. And as we had mentioned that the species blend is unique, within Euglassa dilemma, this manifests in having a species-specific perfume known as a species-specific compound known as HNDB. And in Euglassa viridissima, it has a species-specific compound known as L97. And so at the bottom of the graphs are retention times, and each peak corresponds to a different compound, with the height of the peak corresponding to its relative abundance in the sample. And so we did a decent job of IDing, and after confirmation of that, we could then bring the perfume to South Florida, where we collaborate with the University of Florida in Fort Lauderdale, choice experiments. And so this is what it looks like on the ground in these 10 meter by 10 meter cages, where bees have access to pollen and nectar. And the only thing left to do is acquire naive bees. And so before we get started with experiments, and we've had nest boxes out throughout South Florida in different regional parks. The bees are naturally cavity nesters, in which they nest in tree hole cavities. And so in Florida, we put out wooden boxes that are like pretty note card sizey, and the females will readily colonize them. And so on the left, you'll see a brood box with a tagged female, and those are her brood cells. And then I spent a summer trying to rear bees in the lab, and it was really remarkable watching them grow. And after a bee emerges, we can then use him for the mate choice experiments, or she for the mate choice experiments. And so actively running those looks like inserting Euglassa dilemma perfume in a naive male's leg, inserting Euglassa viridissima perfume into a naive male's leg, and importantly, we're using Euglassa dilemma bees. So the only thing that's changing here is the perfume itself. And so we put, about a microliter dispersed through both legs, and we mark their thorax and can watch them through binoculars to see who's who. And after they exhibit courtship behavior on the perches, we then introduce females. And we let them hang out in the cage for about a week and for enough time for mating to occur. And then later, we can do spermatheca dissections of the females to see whether or not they mated. And if they did mate, then we could do the parental analysis via microsatellites. And then just confirm with GCMS that they weren't finding any other chemicals in the cage. And so the results of the first kind of trial of running these experiments were interesting. We had a lot of male interaction. And so the males spent more time hanging around themselves than they did courting the females. And so they were really interested in each other's perfume, and they didn't spend as much time exhibiting courtship behavior. So if we had been interested in competition, it would have been a great way to look at that. So we didn't get as high of mating rates as we were hoping compared to, the previous round of doing this with perfume and no perfume. So 5 out of the 27 females mated. And this is what a mated female spermatheca looks like. So there's an outer lining, and then inside is all the sperm. And so it's pretty clear whether or not a female has mated or not. And so we confirmed that 2 of the matings occurred with males that had Euglassa dilemma perfume. And because of the haploid diploidy system, you need to have the offspring be females to do the back crossing. And so the other ones we weren't able to tell. But going forward, we're thinking a no choice test. Marina will be good. And like it changes conceptually things a little bit, but I think we can kind of glean the same information. Instead of looking at relative preference, we're going to now look at more of a higher level absolute preference in which, compare the rates of matings between bees that had Euglassa dilemma perfume, bees that had Euglassa viridissima perfume. And this way we can avoid, the male-male interaction. And so I stayed a little while after we wrapped up our 2 choice tests to see if in fact we would see higher rates of mating. And 5 out of the 11 females did mate compared to 5 out of 27. So that's looking better. A little caveat to this is that it may underestimate preference. And so we'll keep that in mind, and that, when you put organisms in a place where they only have one option, they may just settle, since the cost to not mating is, pretty something to grapple with, I guess. And so it also opens up a nice avenue for us to look at the question of local adaptation. We're taking perfume from Mexico and we're using bees in Florida. And so, though the hypothesis is that the species specific perfume is what would elicit mating, maybe the bees have been there for 20 years and there are subtle differences between the 2. So we can now add this 3rd treatment group of Euglassa dilemma perfume from Florida. And so hypothetically, results could look something like this, in which we have Florida Euglassa dilemma, Yucatan Euglassa dilemma, and Yucatan Euglassa viridissima. And if if our prediction is correct, we'd see high rates of mating for males that had the perfume from Euglassa dilemma with females that are Euglassa dilemma. And to just for a little bit of the differences between the perfume collected in Mexico and that of South Florida, almost all the perfumes that we collected in Mexico had this compound benzyl benzoate, which is found in agave flowers and hyacinths and some medications, and they were absent in perfume collected from South Florida. Whereas in South Florida, the bees collect this herbicide and are attracted to this herbicide tricloper that they don't collect in Mexico. And so it'll kind of be interesting to see at least, what the results of this research question are. And on the topic of like subtle differences within a species perfume, that leads to our next question of differences in perfume composition and complexity among, males of the same species. How does that influence mate choice? And again, it's really powerful that we can manipulate these signals in specific ways and dissect the signal up. So going back to the cartoon, we have all of these senders and we have choosers, and each of these quarters has a different perfume bouquet in which they have this species-specific compound, HNEB, but they also have all these other compounds in their perfume, whether or not they were intentionally collected them or if they're kind of, accidents of just while collecting this compound that they were interested in, flowers produce all these other compounds that are part of their headspace. And so they collect all these other things as well. And so there's a lot of variation within a species, but there's more variation between. And so there's this idea in the mate choice literature of hierarchical traits, those being one in which a trait is necessary and sufficient, in this case, to elicit mating, And the other is neither necessary nor sufficient to elicit mating, but enhances preference if the first trait is present. And I think this might be what's going on with Euglassa dilemma. And so we can walk through this question quite quickly because methodologically things are pretty similar to the first research question, in which it involves collecting perfume from males in South Florida and then dividing perfumes into different treatments. And here the treatments would be the species-specific perfume plus accessory compounds. So everything else that's not HNDB that the bees collect, HNDB, and just the accessory compounds. And this can be done synthetically since we've paired up with, we've collaborated with a group in Germany that's able to synthesize HNDB. And then we could also compare the mating rates across these different treatment groups. And so we'll also walk through this one by one of the potential outcomes. So on the top, we have the different treatments. And on the left, we have the potential outcomes in which if, we're assuming that hierarchical signals are the way that this perfume works, in which HNDB is necessary and sufficient to elicit mating, and the accessories enhance preference for it, we'd expect high rates of mating from the males that have HNDB and accessory compounds in their bouquet. We're not going to get 100% mating, but this is all relative. And so we'd also see then that if males have only HNDB, that there'd be some degree of mating and interest from females. Whereas if males have accessory compounds, there wouldn't be any interest. And this is our working hypothesis. But alternatively, perhaps HNDB is the only signal that matters. And in this case, in the no choice matings, we'd see interest in females that are equal between HNDB, males that have HNDB and accessories, as well as males that just have bouquets comprised of HNDB. And no interest in males that have only accessory compounds in their perfume. And lastly, we could also imagine that accessory compounds are the only things that females are interested in. Just because males are attracted to HNDB and collect it, maybe that doesn't mean females are. And so then you'd have the reverse option in which there'd be a high degree of mating then between males that have HNDB and accessories and those that have accessories, but not between the males that present bouquets of just HNDB. And so this is what we think is happening and can be visualized by something hypothetically in which we'd have results that look like this, of high degrees of mating between males that present bouquets of HNDB and accessories, some degree of mating between males that present bouquets of HNDB, and very low degrees of mating between males that lack HNDB and only have accessory compounds. And so this whole time, we've kind of been talking about preference on the female side of things, of what they like or don't like. It's very hard to assign valence of just what they like versus what they're repulsed by. But there's also an interesting flip side of the males themselves are spending a lot of metabolic energy collecting all these different compounds to begin with, and likely their individual history of collection as well as preference plays a role here in the evolution of these bees. And so to go back to our cartoon of different quarters, they're spending all this time collecting different compounds from the environment that usually will always include their species-specific compound. And it's been noted that, throughout the distribution of them, they converge on these species-specific perfumes, but also impressively enough, throughout seasonality, this type of signal is maintained. And so this is work that a former postdoc in our lab, Kathy Dara, had done, in which she looked at 3 species, relatives of the ones that we study in Costa Rica, Euglassa flammea, Euglassa imperialis, and Euglassa tridentata, and looked at their seasonality. So on the left, you can see an NMDS plot, which we're just looking at 3D perfume space, and they're all colored according to their species. And so though there is variation within a species, there's far more variation between species. And then on the right, we're looking at from January to December, the amount of a variation of perfume among individuals of each species. And so it's impressively constant despite vast differences between dry and rainy seasons. And so this kind of relates to, the availability of these compounds in different resources. And so in addition to flowers, males also collect from, detritus in South Florida, clove cigarettes, and populations that maybe don't have as much options to choose from, like island populations, basil, and you can see a lot of them are there collecting. These are all photos from iNaturalist, which is really neat. And so, I'm really interested in, and I think a lot of us working with orchid bees are really interested in how they converge on this type of, stable signal. And so to answer my research question about looking at different individual males that are offered perfume, perfume components that are very stable, I'm going to allow them to collect from chemical baits in a flight cage, the ones that I showed previously, and take repeated measurements over a course of 15 days. I'll take 5 measurements from the legs, and then conduct within and between individual variation covariances and visualize this similarly in NMDS space to compare with wild type bees. And so what's nice about using the chemical baits is it's very rare for a bee to come across a pure compound. Normally they come across, flowers or other environmental sources that are very complex, and they're collecting all these things that, in addition to maybe the compound that they're interested, are sort of a bycatch. But here we can use pure compounds and also concentrations that are unrealistic. And then there becomes this interesting question of what do the bees do then in regards to, how if they had unlimited access, would their perfume look different, so to speak? And so the baits that we'll be using are their species-specific compound HNDB, eugenol, DMB, benzylcinnamate, the herbicide that we had talked about, and a floral compound beta-ocimene, and putting them in different parts of the cage and allowing bees to collect from these. And we could sample over the course of about the 2-week period how their perfume micro evolves and see what happens. And so I'll be looking at the within variation, within individual variation. So how much does the perfume change within a male over the course of the time period, and between individual variation, how much differences between males over the time period happens with a multivariate model that I've put a verbal model here for. And so the phenotypic attribute here is the perfume composed of different, all the 5 chemicals. And then we'll have also a cage effect in case there's something interesting about our cage 1 that gets more wind than cage 3. And so a visualization of what we might see, though extreme, I don't think we will, would be all bees clustering by day. And so the colors correspond to the day of sampling. We'll have 5 sampling days, and the different shapes correspond to different bee individuals. And so an extreme example that you can imagine is the bees have very predictable trajectories, so to speak, in which all bees start on this one compound, and then the next time I sample, they will have all spent this time on this other compound and move on, so on and so forth. And so the biological interpretation would be that they have this very programmed way of collecting and of attraction, innate attraction. On the flip side, you can imagine that maybe they cluster by individual, of the preferences being so extreme that one male only hangs out on this one compound for the whole 2-week period. And so again, these are kind of similar cartoons of NMDS space that are just on 2D space instead of 3D. And so then we'd have, for this instance, the male just staying still in the perfume space. And maybe what is more realistic of what we think could happen is this phenomenon of, again, having this innate preference in which all bees start out on one compound here in the blue, And then over time, their successive visitations maybe affect their attraction to different compounds in sort of a step-like way of we all start on eugenol and then square B goes over and collects, DMB, and then that, affects his affinity for other compounds in the cage so that they diverge over time. And, my hypothesis is that they'll converge. And so you can imagine maybe bees all starting on different compounds in the cage because I put up this bait and one was hanging out here, and then over time converging on a perfume, on a sort of stable area. And a last option that I'm not showing, but would be easy enough to imagine is it's all random and the shapes are everywhere. And, there is no pattern to pick up. And afterwards, similar to what our postdoc had done, we can look at, the amount of change that has happened in a given time. So here, this is for the seasonality project that I introduced at the beginning of the question, in which she looked at the amount of change within a species over the course of the year. And here I'd be looking within individuals over the course of time and you kind of summing up those length and magnitudes of the vectors before. And so I'm really excited for all of these different projects and seeing what we find out with the bees. It's a really neat system to be a part of. And so I want to say thank you all for going through the different questions, working our way down from between species to within species to, individual levels. And I'd be happy to take any questions after. I want to acknowledge everybody who's helped me out with this so far. So thank you, Santiago, for all the help and in our lab. We have such a really great lab, and I really want to thank my QE committee, Phil Ward, Kate Laskowski, Gail Petroselli, Neil Williams, and Rick Carbon for helping me out with a lot of the experimental design and some, conceptual things to think about throughout the process, and my collaborators, the funding sources, as well as, our great department here at EVE and my cohort. So thank you. Yeah.
Do you think that, like within a population, more related individuals would have like a preference away from like their own perfume or towards similar perfumes to themselves?
Yeah, I guess there's kind of this tension of you want to stand out, to potential mates, but you also want to signify I'm one of you, recognize me. And so there's this kind of back and forth. I think potentially with the third research question, we have some ideas of maybe controlling for nest and seeing if this kind of, preference is heritable. We can look at track the nest boxes that we collect from individuals from that. And so I think it'd be really cool to see. Yeah. Thanks for the question. Yeah, Lexi.
I have a couple of questions about like supply and demand here. So like I was noticing as you mentioned that they don't often find the compounds they want in isolation. But like, do you have any sense of which ones they need in high abundance and how abundant they are generally in the ecosystem? And is that at all informed by the fact that they seem to be kind of into compounds that are new and possibly dangerous to them?
Yeah, I don't know about the trichopher, but
Yeah.
like it is, the fact that they're attracted to it is very interesting. So we actually don't know where they get their species specific compound HNDB from. We don't think it's from flowers. We think it may be fungi or microbe oriented. But the other compounds that are, at least for the accessory experiment that I'll be doing in the second research question, we're using like kind of the top 4 or 5 most at least abundant in the perfumes that we sample. And so a lot of those are compounds that flowers produce, and a lot of flowers have a lot of similarities in terms of producing similar from different biosynthetic pathways, the same compounds. And so Jason could speak a lot to this, but things like alpha pinene and beta ocimene are quite common in floral flowers. Did you have another question?
No,
Okay. Yeah.
I was wondering for your first question, if you'd thought about doing like a mating where, like a mating choice assay where you paired females with a male with no perfume and a male with the other species of perfume.
Whether they'd settle. for-
Yeah, whether they would settle for like the no perfume or if they would rather mate
Like,
with.
I'd rather not, yeah.
Yeah, rather have a non-preferable perfume or they wouldn't mate at all.
Yeah, we haven't done that yet. That would be interesting to see whether or not they, you know, for a while, we're just thinking perhaps having a perfume would be of interest. So yeah, we haven't done that. That'd be something cool to explore.
Yeah, I have a question about kind of the species specific compounds that you find. Is it in the other species, are they also seem to be derived from like fungal origins? Or is it kind of different where they might be depending on the species?
I guess to be clear, we don't know that HNZB comes from fungi.
Right.
This is just like something that we think might be happening. And because we see, at least for Ecology, the bees collect from detritus things, like they often will be collecting in leaf litter, that maybe it's something related to small microbes. And for other species, they collect a lot of the same compounds. A lot more of them collect sineal than the Ecology is super attracted to, but I don't know about the fungi. One of our collaborators just found from a big Ascomyda, I think I want to say, one of those. Peter, you gotta help me out. What the one
Big
that,
shell fungus.
yeah, the shelf fungus that at least some Ulema were collecting from that. So that's pretty cool. Yeah.
Do you know what happens when you do a choice experiment? Like you had the males with the two different perfumes, but they were interacting too much with each other. Like what would happen if you could have a setup where they're not able, the males are like separated somehow, but the female is still able to choose, like kind of just based on smell or.
I think I could have like tethering them.
You mean like I'm thinking like like the old experiment, like a Y tube or something. where
Oh,
they
I
can-
see. So I think it'd be tricky because the males, if they don't have a lot of flying space, they won't display. And I think that's kind of been one of these limiting factors for us of like, how much can we restrict the space so that they will display? And they're pretty particular about their perch sites to begin with. So yeah, I guess maybe like a Y-tube assay of just like attraction to the perfumes rather than like a confirmation of mating you're imagining. Yeah, haven't done it, but that could be neat.
I'm just curious about, have you tried sniffing these compounds and can you tell the differences?
You can, like, some of them are pretty strong, like when the bees visit basil, like you smell it. It's pretty neat. And some of the ones that we collected in Mexico that had like a really strong orange tint smelled almost like piney, and that was quite nice.
Well, you can describe all the DMB and eugenol.
Yeah, like the baits that we used to are also like really strong. Like if you just get a little bit on your hands, like a bunch of bees will will come. And I've heard of stories that our postdoc Nick, who was in Florida for a while, had told us of like people using kind of eucalyptal based mosquito repellent and like bees following like kids. And like a lot of, the males don't sting, which is why we can handle them. But a lot of people don't know that, right? And so like when you have your kid wearing eucalyptal based mosquito repellent, all these bees come, it could be maybe a little bit startling. Yeah.
Any other questions? If not, let's thank Marissa again.